morning. Thank you very much for the invitation, Emmett and the OIS team. We're very excited to be here. It's our first time at OIS presenting Adverum. So we are a gene therapy company. We are in the clinic with a program in neovascular AMD, for which we're using a vector called Adverum 022, which is basically a vector encoding aflibercept, one of the standard of care molecules used in the clinic today for those disorders. We have announced that we will present our first cohort, 6 patients, data at Retina Society. We're currently finishing collecting the data for this. Our platform is based on adeno-associated viruses, for which we have a lot of expertise. We know how to design the vectors and manufacture them, which is really important when we're in this business. We are funded, well capitalized to conduct our trials and lead our activities until 2021. And we have a team of experts with us marching along the beat for the company to get where we need to be. As I said, we have our lead program is OPTIC, neovascular wet AMD. We also have a couple of rare disease indications that we're going after. I'm not going to highlight this today. Just to say that we're also looking at other indications in the eye for which the Adverum 022 vector would be beneficial delivering aflibercept. Also to highlight that we have 2 ongoing collaborations, one with Regneron, one with Editas, in the ophthalmology space. We're taking advantage for them to take advantage of our vector technology. So really the premise of a gene therapy for wet AMD is very simple. We all know about the burden of the treatment, the frequent injection that patients have to go under to maintain their vision and keep the disease at bay. So really delivering something that would be stable, and there's a lot of activities out there in different companies trying to extend the durability of action of those anti-VEGF molecules. Having a one-and-done approach would be, of course, the best approach. And also the delivery needs to be simple, needs to be straightforward. And the intravitreal approach, which is really what our technology provides, is in our eyes at least the way to go. We have a proprietary vector called AAV7M8, which has been shown to have improved transduction efficiency in the intraocular space, the retina, but also other areas within the intraocular space, to be delivering that the transgene, the cDNA of aflibercept, and pumping that molecule for it to make its way back to the retina and prevent VEGF activity. I'm going to highlight today a bunch of preclinical studies that we conducted that led us to the clinical trial OPTIC that we'll also talk about a little bit later in my talk. So really, we were aware that we're not the first one trying to do wet AMD gene therapy with a delivery, even intravitreal. So we really wanted to have a strong, very robust preclinical package to answer as many questions as possible. The first one being efficacy. I mean, does this vector work? And so we had a preclinical study in non-human primates where we, in the laser-induced neovascularization model in the choroid, where we had a group of animals receiving just saline 13 months prior to the lasering. And then we compared that to a group of animals receiving the vector also 13 months prior to the lasering. And in the middle, you have here, in the middle of the panel, you have aflibercept recombinant protein administered at the time of lesioning. And basically what you see is that in the presence of the vector or aflibercept recombinant protein, you really have massive abrogation of the grade 4 lesions, which are the hallmark of the model and clinically relevant. This efficacy, so that was 13 months prior, so that you have a hint already at the durability of this technology, of this vector in the eye. And this is represented here where we measured, actually in a different compartment, but here we're showing mostly the vitreous in two groups of animals up to 30 months after the vector being injected. We have this stability of expression as early as 1 month, all the way to 2.5 years. The gray zone that you see that captures the lines is really from another study that we conducted where we had another group of animals that received, we wanted to know how does aflibercept decreased in the non-human primate eye in this model. So we injected a bolus of Eylea in non-human primates, and then we measured in the vitreous the clearance of that molecule over time. And basically what we've shown is that in this case, it was 2 months in the case of the vector-injected animals, but the levels of aflibercept derived from Adverum 022 are equivalent to what's left of Eylea after 4 weeks post-bolus injection. And those levels are maintained all the way, as I said, to 2.5 years. So that's really encouraging to have that equivalence. And we all know that aflibercept is believed to be active, 6 to 8 weeks, maybe even longer in some patients. So having an equivalent at 4 weeks is really, really encouraging. There's also a question that we had around safety. What does happen to the retina, even a naive retina, in the presence of a molecule that suppresses VEGF? Is there going to be any kind of mischief on the retina itself? And basically what we show here is that in the animal that was conducted all the way to 13 months, 2.5 years, there's really no change in the retinal structures as observed by OCT. We also looked at the function of the retina in that animal. Again, 30 months, 23 or 30, 19 months or 30 months post injection of the vector. And by ERG, what we see on the left panel is with increased intensity in a normal ERG. On the right panel, you have a multifocal ERG to look at different components of the retina. And basically what we see is that there is no alteration of function of the retina in those animals. Again, up to 30 months post-injection of the vector. I have a bunch of preclinical data that I could not, because of sake of time, I could not present here, but rest assured that we've demonstrated safety, even in the presence of neutralizing antibodies to the vector. That's a question that we asked a lot, and I'll be happy to answer any question that you have after the talk. Just in terms of the clinical trial, the OPTIC study, so it's a study that's a 2-year study, but with a readout at 6 months. The patients have to be responsive to an anti-VEGF, either Lucentis or Eylea, and they are given a taper of a course of steroid at the time of administration of the vector to ward off any kind of acute inflammation. It was originally, so the primary endpoint, of course, is safety. The secondary endpoint are BCVA, number of rescue injection, as well as CRT, as measured by OCT. And those are the data that will be presented, as I said, at the Retina Society in September. Initially, it was a dose escalation. We started at 6 × 10^11. In observing the clinical data, we announced that we have seen robust preliminary efficacy in terms of anatomical response, and therefore we decided to dose down to a lower cohort, and nothing to do with safety, as the DMC allowed us to move on to the second highest dose initially, but we decided to go down. I'm almost finished, 20 seconds. Just as I said, stay tuned for our release of the data in September, and we will provide other updates as we move forward. Thank you very much for your attention.